Hey everyone, and thanks for joining me today for episode 826 of People Behind the Science. I'm your host, Dr. Marie McNeely, and I'm looking forward to speaking with our guest scientist, Dr. Beth Stroupe, today. This episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And listeners, Beth has some wonderful stories to share with all of us in our episode today, so get ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Beth Stroupe. So, Beth, welcome to the show today. How are you?
I'm doing well. Thank you so much for your invitation. This is my first podcast, so I'm very excited to be talking about my work.
Well, Beth, we are thrilled to have you with us and to learn more about you and your work today. But let me first give our listeners a sense of your current position and how you got there. So listeners, Beth is professor in the Department of Biological Science and the Institute of Molecular Biophysics at Florida State University, or FSU. She's also director of the Molecular Biophysics Graduate Program there. Beth completed her undergraduate training at Wake Forest University, where she majored in chemistry and minored in music. She was then awarded her PhD in biochemistry from the Scripps Research Institute. Afterwards, Beth conducted postdoctoral research at Brandeis University before joining the faculty at FSU, where she is today. And Beth, we're excited to hear the story of your path in science and also your life outside the lab. So can you share with us, what do you like to do when you're not very busy doing science?
So my partner Brian and I like to hang out with our two cats, who are named Oscilla and Wasissa.
Aww.
And they're named after two of the local dark water rivers that Brian and I like to kayak on in our free time. We leave the cats at home. We also have a big garden, mostly in our front yard, so that keeps us busy. We also like to hike and just explore the great outdoors. Tallahassee is in the panhandle of Florida, so we're a little more rural than the stereotypical Florida of Disney World or Miami.
Well, I love it. It sounds like you've got a lot to keep you busy outside of your research, but you mentioned that you garden. What do you like to grow in the garden?
So the major gardening seasons in North Florida are actually in the winter. In the summer, it gets a little too hot and buggy. So we like to grow anything cruciferous. So collard greens or mustard greens, broccoli, cabbage, cauliflower. In the spring, you can grow beans and squash. And in the early summer, you can grow peas. Black-eyed peas are our particular favorites.
Oh, that's awesome. Well, I love to hear more about your life outside the lab, but I know you were doing some remarkable work in the lab as well. So let's chat about that next, Beth. How do you describe your work to people who maybe aren't familiar with your fields of structural biology and biochemistry? And how do you describe your work to people who might be outside of science altogether?
So our bodies are made up of chemicals, biomolecules. We've all heard of DNA and RNA and proteins. And the research in my lab and the folks who work with me are interested in understanding what these molecules look like so that we can understand how they work. And I'm a very visual person, so I like to see things so that I can understand how they're connected and what makes them tick. But doing that with biomolecules is particularly challenging because they're very, very tiny. And we have to use different techniques to do that. One of the techniques that we use is to coax the molecules into forming three-dimensional crystals so that we can amplify their tiny pieces to make them larger so that we can visualize them. Another technique that we use in my laboratory is called cryogenic electron microscopy, or cryo-EM. And in this technique, you immobilize the molecules on very tiny, thin grids and image them in the electron microscope. And then you use computers to put them back together into 3 dimensions so that you can see what they look like literally at the atomic level.
Oh, I think this is so cool. And can you share with us why it's important to be able to see what these molecules look like at the molecular level?
Well, one is just basic curiosity. If you want to understand how things work, one way of doing that is to take them apart and put them back together again. You can't do that if you can't see them. But from a more practical point of view, if you're interested in controlling those molecules, say to develop a drug that might control cell division, for example, and be involved in, say, the cancer disease phenotype, one approach is to visualize what that molecule looks like so that you understand how it works. So you can throw a monkey wrench into it to either break it or throw in some enhancer to make that molecule work better if the molecule is broken and is causing the disease. So this really gives us a fundamental chemical understanding of how biology works so that we can develop mechanisms for controlling it.
Well, Beth, I think the research that you're doing is so cool. And I think being able to work on really exciting research projects can be motivational in and of itself. But I think in science, as well as in other fields, you often need a little boost of motivation or inspiration to keep you going when times are tough. So do you have a favorite quote or a saying or just something that really motivates and inspires you?
So there are a lot of people who are wiser than me and have said things that motivate me when I feel like I need a little push to get into the lab or to go into the classroom. One of my favorite quotes is from Adam Gopnik, a May 10th, 2004 issue of the New Yorker magazine. And he was writing about a youth football coach, but I think his words can inspire those of us who want to teach others how to do scientific research and how to understand basic science. So Gopnik said, "It is said sometimes that the great teachers and mentors, the rabbis and gurus, achieve their ends by inducting the disciple into a kind of secret circle of knowledge and belief, making their charisma a kind of gift. I suspect that the best teachers do something else. They don't mystify the work, but insists that all the magic they have to offer is a commitment to repetition and perseverance. The great oracles may enthrall, but the really great teachers demystify." I don't know that I'm always a great teacher who demystifies, but it does give me something to aim for.
Oh, absolutely. And I think there are so many mysteries that remain in science. Being able to kind of break those problems and questions apart and help people understand how these things work can be really motivating and inspiring.
I agree.
Well, let's talk about some of the people who you've worked with over the course of your career who might have motivated and inspired you. I think as a scientist, you get to work with some remarkable people, these role models, mentors, and just inspirational figures that you encounter. So are there particular individuals who rise to the top of your mind as people who've been really impactful in your career?
So my PhD mentor was Elizabeth Getzoff, and her PhD mentor was Jane Richardson. And both of these women have really inspired me because they were able to enter into the field of structural biology before it was the more general field that it is now. And they really used their own unique perspective and their own unique skills to develop methodologies for visualizing these biological structures. So Dr. Richardson, for example, invented the ribbon diagram, which is a general way that researchers use to look at sort of cartoon versions of protein structures. And she did this in the 1970s and 80s when her husband was doing X-ray crystallography at Duke University. And she wanted an easier way of looking at things. And of course, this was before we had the graphics that we do on contemporary computers. And so she was able to look at his representations of the protein molecules and draw by hand this cartoon depiction of the molecules so that we could understand intuitively what they look like. And now, of course, we have computer programs that do it for us. But Dr. Richardson took her artistic ability and combined it with a scientific understanding and really had a novel way of visualizing protein structure. And my PhD mentor learned from her how to think about proteins, not just in terms of their chemistry, but as these large macromolecular machines that do interesting chemistry that it's a little bit harder to do sometimes in the laboratory, in an organic lab, doing it synthetically, where biology has evolved these complex chemicals that can do a range of chemistries that we humans aren't quite as good at doing.
Definitely. And it sounds like, Beth, you had these fantastic influences on just the way you thought about your field relatively early in your career, which I think is very helpful in many cases. But perhaps we could take it even a little bit further back. Can you share with us how you first got interested in science and maybe how you started thinking about wanting to pursue structural biology or biochemistry?
So I was very lucky in that I was raised in a very supportive and loving family. And my father was an analytical biochemist, and my mother was a mathematician statistician. We like to joke that if you took a Mendelian cross of an analytical biochemist and a statistician, you end up with a structural biologist, because, of course, we use biochemistry and mathematics to understand what these molecules look like. Mm-hmm. I became interested in particular in metalloenzyme chemistry. So in the biological chemistry that is performed by enzymes that use metal ions to enhance their chemicals, because I took inorganic chemistry and biochemistry at the same time when I was an undergraduate and realized that the source of everything on Earth comes from the interaction of the sun with a metal-containing molecule, chlorophyll, that allows us to turn sunlight into energy. So I became fascinated at this intersection between inorganic chemistry and biochemistry, and the rest, I guess, is history.
I love that. So you started getting interested in science. You kind of had this idea that being an academic faculty member was an option because of the positions your parents had. Can you maybe talk about what are some of these key moments that really helped you connect the dots and get to this faculty position?
I think one of the most formative experiences I had was when I took organic chemistry when I was an undergraduate. And I had a really excellent teacher at the time, and he taught us how to think about the problems. So we didn't just have to memorize things. We could actually figure out the organic chemical reaction by understanding the pathways of electrons. And the first time I realized I could figure stuff out from early principles of maybe not basic principles, but from understanding the underlying processes was the first time that I thought, yeah, I think I can do that for a career and have an independent research project of my own.
So then what were some of these steps that you took then? We mentioned that as an undergraduate student, you were there at Wake Forest University. How did you decide you wanted to go to the Scripps Research Institute for graduate school?
Like I said, I sort of had this bioinorganic interest develop a little bit late in my undergraduate career. So sort of halfway through the process of applying for graduate schools, I switched half of my applications from straight up chemistry programs to biochemistry programs. And of course, I recognized that Scripps was a leader in structural biology of metalloenzymes. So my interests overlapped really strongly with a bunch of different people who were at Scripps at the time. So it just was a really good fit between my interests and the opportunities that were available.
Definitely. And we mentioned in our introduction that as an undergraduate student, you also minored in music. Can you share a little bit more about this interest in music of yours?
So I play the flute, not as much as I used to, not as much as I wish I did. But yeah, I started playing in the 4th grade when the music program in my public education started, and played all the way through college. And then I took a little break during graduate school. But then as a postdoc, I started playing again with one of the local music groups in the Boston area.
Do you still play today?
Not as much as I wish I did.
I'm a violinist. I have to ask. My violin has been gathering dust, I will say, in recent years. So it's difficult to kind of pick that up every now and then.
Yes.
Well, it's wonderful to hear a little bit about these early stages of your academic training. We mentioned you went to Scripps Research Institute for your PhD. You then were at the crossroads that I think a lot of students find themselves at as they're finishing graduate school, kind of what to do next and what path to take. How did you decide where you wanted to do your postdoc?
When I was finishing up graduate school, I learned X-ray crystallography as a graduate student. And I was fortunate to be at Scripps because Scripps was one of the institutions that was really at the forefront of the three-dimensional electron microscopy field in the early 2000s. So it was on my radar, I think, before it was a commonly acknowledged structural biology technique. So I sort of saw where things were headed and that it was going to emerge not as this niche technique, which it was in the early 2000s, but that it was going to become more mainstream. And so I did decide that I was going to move from X-ray crystallography into cryo-EM. And again, at the time, the field was fairly small. And so there were only select places in the United States that one could learn this technique. Brandeis is also one of the leaders in three-dimensional cryogenic electron microscopy. And I was happy to move to Boston to spend some time up in the Northeast. So I guess my choice was to learn a different structural technique that I could then apply to the metalloenzyme system that I was interested in.
Absolutely. I think that's a smart strategy to build these complementary skill sets that then will allow you to launch your independent research laboratory. And can you share with us then how you ended up at Florida State University? This was the first place you landed for your faculty position, and it sounds like it's been a good fit as you are still there today.
Yeah. So even through the 2000s, up until 2009, when I started at Florida State, cryo-EM was still in its early stages of broadly being adopted as a structural biology technique. And the way that it went from being this niche technique into a mainstream structural technique is that the technology kind of had to catch up with our ideas for what we wanted to learn from the structural biology approach. So during the early 2000s, the companies that built electron microscopes got better at building stable cryo stages so that you could take really high resolution pictures of the molecules without them moving around. Because if stuff moves while you're trying to take its picture, it's going to smear out and then you won't get a high resolution image of it. So the microscopes became more stable. The imaging platform, the camera that we used to take the pictures became better, just higher resolution, more accurate. So we could get really detailed pictures of these molecules. And the computers caught up with it, right? So you need a big, huge GPU in order to do the calculations that you need to take these two-dimensional pictures and turn them back into a three-dimensional image of the molecule. And this transformation happened, again, from the early 2000s into the 2010s. And Florida State happened to be an early adopter of the cryo-EM technique. We were one of the first institutions to get a Titan Krios, which at the time was made by the company FEI. It's now made by Thermo Scientific. So FSU really was at the vanguard of this technology in 2009. And I was lucky that they offered me a position. And Florida State also has some other great collaborative opportunities. So FSU is also home to the National High Magnetic Field Laboratory. So I've been fortunate to work with them. We also have a collaboration with Oak Ridge National Laboratory. So I've developed a collaboration with them as well, as we have tried to do cryo-EM on a sample that was rather recalcitrant to the technique. So we had to take orthogonal approaches through these other collaborations that were facilitated by the university.
Well, Beth, it sounds like you have a wonderful research environment for the kind of work that you're doing today, but let's dive into some of the detail of your research. Do you have a particular project at the moment that you're working on, or a recent one that you are just so excited about and want to tell us more about today?
I won't say that this is a recent project because it has been on my radar since I was a graduate student at Scripps. Mm-hmm. In fact, this project was supposed to be my PhD project, and I failed miserably at it.
Oh, no.
For a number of reasons. First of all, cryo-EM wasn't as developed as it is now, so the technology simply wasn't there. And we just weren't as good at making the enzyme that I'm interested in. So the enzyme that I'm interested in is called sulfite reductase. And it has two different protein components that come together to reduce sulfur from sulfite into sulfide. And sulfide is the version of sulfur, or the redox state of sulfur, that's found in your protein molecules and other small molecule cofactors that are found throughout the cell to do all sorts of chemistry. And this chemistry is rather tricky to perform because it's a 6-electron reduction. Most electron transfer reactions are 1 or 2 electrons. 6 electrons is an awful lot. And as I mentioned before, humans aren't always as good at doing chemistry as nature is. So the analogous chemistry is really difficult to do synthetically. And when you hear about nitrogen fertilizer plants catching on fire, it's because that chemistry is also a 6-electron reduction reaction. And humans are really bad at doing that kind of chemistry. Mother Nature is a lot more clever. So when I joined Dr. Getzoff's lab at Scripps, they had just solved the structure of the iron-containing subunit, and they were the first lab to achieve that result. So my project was supposed to be to understand how the iron-containing subunit interacted with the flavin-containing subunit to perform this really amazing chemistry. And like I said, I failed rather miserably. And it has taken about 30 years for technology and biochemistry to come together so that we could determine the structure of just these two components talking to one another so that we could understand the basic assembly unit for the larger complex. The next big step is to determine the structure of the whole complex, which we call the holoenzyme or the whole enzyme. So there's 8 copies of one subunit and 4 copies of the other subunit. So it's a dodecamer. There's 12 subunits. And the subunits that form the octamer part of the molecule are really, really flexible. We think about it like it's an octopus. And this is really fascinating because it's these movements that allow the molecule to perform the chemistry that it does. But it's really hard for structural biologists because we need our molecules to sit still and all be in the same conformation so that we can amplify their tiny, tiny bonds so that we can visualize them on our computers and in three-dimensional space as we understand it. So if things are moving around, if things are really flexible, it's hard to get them all in the same position. We've gotten really good at doing biochemistry to understand how these motions work to do the chemistry that they do. And hopefully in the next couple of years, we'll figure out what this whole complex looks like.
Oh, this is really cool. And it sounds like this project has been a long time in the making. What does it feel like for you to be at this precipice where it sounds like you might have a major breakthrough very soon?
Well, we'd like to think that we already have had a major breakthrough in just solving this one small part of it, because that in and of itself took us a really long time because we had to not only figure out the biochemistry to get the molecule to sit still, we also had to figure out some tricks in order to work with the process of cryogenically preserving the sample in order to get it to stay intact in the really thin film of liquid ice that you need to image it in the electron microscope. Nevertheless, I think we're close to getting the whole complex, and I have some really talented graduate students in the lab right now who are doing the biochemistry that we need to make sure that we've got the right version of the molecule to coax it into submission, as it were, so that we can image it and reconstruct it in 3 dimensions.
Well, Beth, this is so exciting, and listeners will definitely have to keep an eye out for upcoming papers coming from Beth's lab to see more new developments from this project. And to make important progress in science, I know it's critical for researchers to use high-quality lab products and supplies. And we'd like to take a moment to thank our sponsor, Innovative Research, and talk about their portfolio of chemiluminescence immunoassay kits, which offer high sensitivity, specificity, and reproducibility. These kits are designed for research targeting diagnostics, pharmaceuticals, or forensics, as well as environmental monitoring, food safety, and agricultural research. Visit www.innov-research.com. That's www.innov-research.com to view their entire line of chemiluminescence immunoassay kits. Now, returning to our conversation with Beth, this recent project that we were talking about highlighted both successes and struggles, and I think science has a lot of both. There are a lot of difficult times, but also a lot of really exciting moments. So let's start with the challenges and failures, because everybody goes through them, but not a lot of people talk about them. So, Beth, do you have an example of a time that you really struggled with something in science, or maybe you had a major failure? And if so, can you walk us through how you get through these tough times?
I firmly believe that failure is part of the process. Especially when you're a student, you learn when things don't work. If everything is smooth sailing, you don't learn how to troubleshoot and you don't learn how to figure things out. And figuring things out is part of figuring out how they work, which is, of course, the goal of a structural biology approach to understanding biochemistry. This is not easy to practice because it does involve a lot of stuff not working, but it makes the successes all the more exciting. It's also, I think, really important to understand when you are failing productively, that is, you're learning from your mistakes, and when you are failing non-productively and you need to find someone to help you, someone who has done it before or someone who knows more about it or has more experience And I have been incredibly lucky that I have had those mentors throughout my career, people who've been willing to listen to my not so intelligent ideas that needed readjusting, as it were, to get me back on track so that we could make successes happen.
Absolutely. Do you have an example maybe of a recent challenge or something where you were failing unproductively and you had to get help?
Well, trying to preserve the whole complex, this dodecameric complex, has been particularly challenging. And almost everyone who's been in my lab, almost all of the students who have passed through my lab, have tried to work on this project. Preserving it cryogenically is called freezing the sample. Initially, we couldn't freeze it at all. And we overcame that challenge by finding a new material that we could use on the chip that we use to freeze the samples. So moving from a carbon surface to a gold surface allowed us to freeze it. And I couldn't explain why. Just trying a bunch of stuff until something works. But even once we were able to get good freezing conditions, the molecule still we can't do the mathematical alignments that we need to do because it is so floppy. Slowly, we are taking the protein apart and trying to make the floppy bits not quite so floppy. And one way we achieved this was by engineering disulfide bridge. So we took 2 amino acids that were not essential to the function of the molecule, and we changed the chemical nature of those residues into cysteines. And cysteines are the amino acids that have sulfur groups attached to them. And these sulfurs can form covalent bonds with one another to lock a protein into one conformation. So figuring out how to do that has locked the protein down. So parts of it that used to flop around are now sitting still so we can take pictures of them. It's also told us something about the chemistry that's taking place, though, because we've shown that the electrons don't just transfer from one polypeptide to the closest polypeptide, but rather they can move across the molecule, moving from one subunit to a subunit that's attached to a different polypeptide that's part of the complex. I guess my point is you never know what you're going to learn from doing an experiment. So we were just aiming to do some structural modifications to make the molecule sit still, but we learned something about the chemistry along the way.
Well, Beth, I love this story. And I think this is such a common situation in science where as you solve one challenge, a new one emerges and you just have to keep pulling away, removing one of those hurdles at a time until you get to the solution. I think that perseverance and that dogged determination is critical in science.
You can either go for the low-hanging fruit or you can just keep boring away at a project. So we are taking the latter approach in my lab.
I love it. Well, we mentioned one of these big wins that your lab has had relatively recently, and I think it's so important to celebrate these successes in science because you're putting in so much work, so much time, so much energy into this research project. So do you have a story of a success that you would like to tell us more about today, whether it was a big win or a small but particularly meaningful one?
Just solving the structure of this dimer between the two subunits. So the subunit that binds the irons and then the subunit that binds the flavins. So in the 1970s, before I was doing research, it was understood that these two subunits bound to one another. And this odd stoichiometry where there's 8 copies of one that bind to 4 copies of the other subunit. It's just kind of odd. When I started my research group, one of the first things we set out to do was to show that this initial asymmetric assembly was incorrect. Biochemistry has advanced since the 1970s. We're so much better at doing what we do. We're going to show that they were wrong. And nothing we have done yet in the lab has convinced me that this initial assessment was incorrect. In science, you can only disprove a hypothesis. Mm-hmm. We have not disproven that hypothesis.
Interesting.
Once we figured out that the people who discovered this enzyme were correct, we started trying to discover what the structure looked like, and everything we did didn't work. Cryo-EM didn't work. X-ray crystallography didn't work. And so we were forced to take these orthogonal approaches. And the first orthogonal approach we used was in collaboration with the National High Magnetic Field Laboratory that, again, is part of FSU, but is a national facility funded by the DOE. And we worked with a group there who was really good at using a technique called hydrogen deuterium exchange to understand which parts of the protein were exposed to the solution and which parts were masked because they were talking to other components. And in doing this, we realized that the two subunits interacted in a really odd way. They don't interact like you would expect given the chemistry that the molecule does. So we showed this biochemically, and we knew this for many, many years, but it wasn't until we were able to use cryo-EM to solve the structure that we could show the detailed atomic resolution basis for this unusual interaction between the subunits. And I know that sounds really down in the weeds and kind of nerdy, and it is, but it's super interesting just because it's unusual.
Oh, absolutely. So what was it like that first time that you saw the pieces fall into place and you could actually finally see this detailed picture of how everything fit together?
It was pretty cool. When you are a nerd and you're interested in geeky things, you just get that tingly feeling when stuff just falls into place. And it did. And I wouldn't have gotten here if it hadn't been for the really talented students in my lab who were able to be persistent when I was feeling down, and I was able to encourage them when they were feeling frustrated. And I've just been really lucky that we've had such a great set of students through my group to do this research with me.
Oh, that's wonderful. I think it is a team effort, absolutely, in science. And you have to celebrate, like we mentioned, the wins. How did you celebrate this win, Beth?
We went out to dinner together, and we have a lab mascot who is a giant pink unicorn. His name is Henry Sparkles, and he comes with us to all of our major lab events and all of our presentations. So he comes to all my students' thesis defenses. He came to my tenure talk when I was presenting to my department to tell them about our research. So Henry Sparkles is always part of our lab celebrations.
Oh, I love that. What was the genesis of Henry Sparkles? How did he become the mascot?
I don't even remember at this point. It's always been there. And when the students are stressed out because they have a big test, they like to sit with him. He wears an FSU t-shirt to remind us where we are. He's a member of the lab in his own right. I think he still has a Facebook page even.
Oh my goodness, listeners, we're going to have to look into this. I think that is remarkable. And I think taking the time to celebrate enjoying these moments with your lab is absolutely important because you're answering some really difficult questions. And I know when you have these successes, it's easy to get pulled into this cycle of wanting to work more and more, longer and longer hours as you get closer to a solution. But we try to encourage listeners to relax, take a step back, perhaps pick up a good book and think about something other than their science for a little while. So we love getting book recommendations from everybody that we have on the show. Beth, do you have a book that you've really enjoyed that you would like to recommend for all of us today, whether it's related to science or not?
So one of my favorite books is by Carson McCullers, and it's The Heart is a Lonely Hunter. And this is a coming of age story of a young woman in the South. And it has some themes that I think are really relevant now. It's not a science book at all, but it deals with the themes of fascism. It deals with themes of underrepresented people trying to find their way in a world that can be confusing and challenging at times, but finding the joy in the in-between moments.
Well, excellent recommendation. You are the first to mention this book, so we will definitely add it to our website for our listeners to find there on our resources page. And one of the other things that's wonderful about life in science, not only do you get to work with great people and do really cool projects, you get to travel the world and meet people from all over the world. So we love talking about these travel opportunities on our show. So Beth, do you have a favorite place that your science has taken you that you can tell us about today?
One of the first international collaborations that I had was with a group in the UK. So I will always cherish the time that I had when I was able to go there to perform some experiments as a graduate student that I wouldn't have been able to perform in my lab at Scripps. So I will always feel kinship with that group of British students and international students who were in that lab, who welcomed me to their space and taught me a lot about how to make unique tetraperols.
So where in the UK was this lab based?
At the time, they were in the outskirts of London, but now they're at Kent University.
Gotcha. And was this your first international trip?
No, I had been to the UK as well as France before, but this was the first time I'd ever really worked in a lab that wasn't in the US.
Very cool. So what was it like getting immersed in that lab, and were there differences and similarities that you saw between the previous places that you worked in there?
At the time, there were more international students there. Certainly, there were international students at Scripps. There were fewer at Wake Forest when I was there in the late 90s. So, yeah, it was interesting to see how those international communities interacted with the students and how they fit in despite the cultural differences.
Oh, that's so cool. And then have you been back to visit the lab? It sounds like perhaps since this first trip there.
Yes, I have. I've been back just to say hi, and they come to the U.S. often for a Gordon conference. So we have reunions there.
That's awesome. I think that is one of the wonderful things about science is these people that you meet over the years. You see them at conferences, you kind of build this network, this community, and almost like this family in science where you get to visit each other at different places and different times.
That is one of the most wonderful things about the way science works in the United States, where you can learn different cultures and appreciate how different communities come together to answer common questions.
Definitely. And were you able to do any sightseeing around at that time, the London-ish area?
Yes, I also spent some time there as an undergraduate. And my grandmother and my great aunt were both big Anglophiles. So I sort of grew up thinking about Britain as a home away from home.
That's lovely. So what are some of your favorite places to go to that listeners must check out if they're in the area?
So I've always liked Wales and the Lake District, which is in the northern part of Great Britain. There's interesting outdoors. There's a lot of hiking that one can do. And it's just really, really a beautiful countryside.
That sounds lovely. Listeners will definitely have to visit the Lake District next time we're over there in the UK near Wales. And I think in science, you get to work with these amazing people, as we mentioned, from all over the world. But there are some amazing characters that you meet in science as well. These funny, creative, quirky people that I think maybe go against the stereotypes that people have about scientists in their white coats being very serious, working in the lab all the time. So we love sharing these human moments in science, whether it's fun lab traditions or just fantastic memories that you've shared with colleagues over the years. So Beth, do you have an example or a story that you can tell us about today that shows this human side of science that goes against the stereotypes?
Where to begin? So one of the more interesting individuals that I was lucky to have met was Don Casper. So Don was one of the, I guess, grandfathers of the structural biology field. And in fact, he coined the phrase structural biology in the 1960s to describe this approach towards understanding the way that biology works on a molecular level. And so Don was at the MRC when Rosalind Franklin and that crew was there figuring out what DNA looked like. So he knew her. And then he had wonderful stories to tell about all the different people that he knew from his time there. He then did a stint at Brandeis, and then he came to Florida State. So in many ways, I've sort of followed Don around. And he just had a way of understanding structural biology that I think sometimes we lose in our technology-driven approach towards it, because he had to think about these molecules and build models of them by hand rather than just asking a computer program to display it and then magically spin it around in 3 dimensions. He really had to think about things sort of at a literal level, coming up with analogies so that we could understand the way in which nature has creatively evolved to solve different biophysical problems.
Well, it sounds like a fantastic opportunity to meet sort of one of the legends of the field. Was it intimidating the first time that you met him?
Don was always incredibly supportive of all of the younger people that he worked with. In particular, he was very supportive of women in science. The biophysical field, structural biology, historically was not a very open field. It was very male-dominated for a very long time. And so Don has always been very supportive of opening up with opportunities for everyone because he loved structural biology so much that he just wanted to share it with all of the people that he met and trained. So yeah, I hope that spirit stays.
Definitely. And it sounds like you can hear your own passion for the field and the work that you're doing coming through as you talk about your work. And I think that in and of itself can inspire others to become interested in the field as well. And we need to bring all of these different perspectives and people into the field to solve some of the difficult questions that you're facing, because I know you have limitations, unfortunately, as a scientist, whether it's funding, staff, the technologies we've sort of alluded to evolving over time, even just the feasibility of the issues you want to address. So if we took away the things that typically hold you back, Beth, what is the one research question or problem that you would want to address first?
I would really love to go back in time when the Earth transitioned from being a sulfur-based environment to an oxygen-based environment, to understand what pressure the enzyme that my lab is interested in, sulfite reductase, to use the cofactors that it uses rather than evolving into a more modern form of cofactors. Sulfite reductase is unique because it has this heme-like moiety called seroheme. So seroheme is a lot like the heme that's in hemoglobin that carries oxygen through your body, but it's more ancient and it existed before the world was an oxygen-rich environment. And for some reason that I'm not sure we'll ever really understand, sulfite reductase stuck with seroheme when other enzymes evolved to use the heme like you find in hemoglobin. And it would be fascinating to understand why this particular chemistry needs that rather bizarre cofactor.
This is a fascinating question. We'll need a time machine first order, right?
Indeed.
Well, Beth, this is a wonderful problem for myself and the listeners to ponder today and just think about how these major changes were happening at the early days of our planet. So we appreciate you bringing this to our attention and dreaming big with us on the show. And I'd love to end our conversation by talking about advice, because listeners out there may be on their own scientific journey or just struggling with different things in their lives. So do you have a piece of advice that somebody gave you at some point that really helped you that you can share with all of them today?
Don't let people get you down.
And how did this help you?
Well, I think there are some people who feel that they need to put you down in order to show how smart they are. And I don't know that that's a really helpful way to go about life, but it's going to happen. So you need to find a way that you can just be like a duck and let it roll off your back, because otherwise the competition will be rather rough. I sort of give myself 24 hours to be upset or disappointed when I get a bad review on a paper or a negative response for a grant. And then I pick myself up, dust myself off, and move on.
Definitely. And I think that's such a helpful approach and way to think about it, because I think this criticism, this sort of peer review, all of this is inherent in science. There are going to be people judging you and your work all the time as a scientist. So you need to find out how to not take it personally.
And that is easier said than done.
Oh, definitely. Well, wonderful piece of advice for listeners. Is there any other last piece of advice, Beth, that you'd like to share, or a last note of inspiration that you would like to end on today?
Just keep moving forward. And especially in the current time, it is challenging to be an academic scientist. And yet it is so important. We just need to keep moving forward, and hopefully the world will catch back up with us.
Certainly. Well, I think this is an important message as well. This perseverance during difficult times is critical for scientists, and particularly critical today. So, Beth, thank you for sharing your stories and insights with us today. If our listeners are intrigued and they would like to learn more about you and your work, what is the best way for them to do so?
If you Google me, you will find my FSU webpage, and from that, you can find my lab webpage. I confess it may not be the most up to date, but you'll be able to find my email address. So I'm always accessible via email, and I'm happy to answer any questions or introduce you to research opportunities at Florida State.
Fantastic. Well, thank you for sharing these resources, Beth. Listeners, definitely take some time to peruse her lab website. Get in touch if you have particular questions. And Beth, it's been such a pleasure to chat with you today. Thank you so much for joining me on the show.
And thank you again for the invitation. And I always like to talk about my rather niche, bizarro interests.
Well, it was wonderful to learn more about you and the work that you do. Listeners, always great to have you here with us as well. We hope you join us again next time for another episode of People Behind the Science.